Hello, everyone, and welcome to the Creation Matters podcast, where we focus on sharing the reliability of the Bible, real science, and the gospel. I am your host, Preston Knapp, and joining me in studio as my co-host is Ashley Coulter. Ashley, thanks for sitting in.
Thanks for having me.
Our very special guest today is Dr. Mark Armitage. He is the senior microscopist with the Dinosaur Soft Tissue Research Institute, and he's also the founder, or one of the founders, and director of the Dinosaur Soft Tissue Research Institute, has a PhD in science education and leadership, and many other degrees also of various fields of study. So, Dr. Mark, thanks for joining our podcast today.
Thank you. And it is an EDD, educational doctorate. PhD is more of a research degree, but I think I've done enough research to qualify for one of those as well. So.
All right. Well, thank you for the clarification there. I appreciate it.
Sure.
All right. So today we're going to be talking about dinosaur bones, and that's kind of something that, here as creationists we like to talk about is dinosaurs, fossils, their bones. But we're going to be talking about something a little bit different today. We're going to be talking about what we find inside these fossils. So, Dr. Mark, you're kind of an expert in this field, so maybe explain kind of what led to us even looking inside dinosaur bones in the first place.
Right. Well, my training in science was very technical. I studied under Dr. Richard Lumsden, who is the dean of the graduate school at Tulane University and an eminent microscopist. That's a person who really knows microscopes, light microscopes, fluorescent microscopes, confocal. Maybe some of your listeners have heard of confocal, but also electron microscopy. And so I got wonderful training. And then I started working in universities. I worked at the Masters University at Azusa Pacific, and my last position was at California State University, Northridge. And I ran a big, beautiful lab there with a bunch of electron microscopes and other systems. But it was great to have that kind of training because I learned all about soft tissue and how to prepare soft tissue for any kind of study. In biology, I would prepare tissues and train professors and other folks to process their tissues. And so I knew a lot about how tissues behave, how they're preserved, how they break down quickly, and, the whole gamut of reagents that have to be used and processes, protocols that have to be adhered to in order to make sure that your tissue is going to look like it's supposed to under the electron microscope. So, because we can introduce artifacts or changes into things that we examine very easily. So when I started reading about Dr. Schweitzer's discovery, and then I went back and studied the work of Roman Pawlicki from the 60s, who's in Poland, I was astonished that they were reporting on soft tissues. And I instinctively knew that they couldn't survive, not even 1,000 years or 1,500 years. It's very rare to find preservation, cellular preservation after that length of time. And so I was stunned and shocked. And so I decided, hey, we're going to go on a dig. And we did. We arranged to have a dig in 2012 in the same dig site in the Hell Creek Formation in Glendive, Montana, that Dr. Schweitzer had recovered the T-Rex femur from. And of course, everybody was looking for femurs at that time. This is back in 2010. So I think she started her work in 2005. But we didn't find a femur. We found the famous 48-inch long Triceratops horn, which is all over the internet. But we found significant world first findings in that. And so since 2012, we've been digging in deeper and deeper to really understand what's going on out there. And we've made tons of world first discoveries, which I'm happy to share. But that's in a nutshell what prompted me to go out there and prove this to myself that was really out there because of my training in soft tissue.
Wow. Amazing.
That is. So, Dr. Mark, tell us what you have found in these bones and what is the whole process, the scientific process of...
Okay. Great question. Well, cells, dinosaur bone cells, collagen, blood veins, blood vessels, these have been known for about 20 years since Dr. Pawlicki and Dr. Schweitzer's work. But when we got involved, we were the first ones to show stretchy fibrillar bone. In other words, bone is made by these little cells that live in your bones, my bones. They actually make a collagen carpet, and then they spread themselves out on that carpet, and they sort of cement themselves in with bone mineral. So they're actually living cells inside these little they're called lacunae. They're little spaces that they create for themselves, and then they create all these little tiny tunnels. They're very narrow, maybe 500 nanometers wide, and that's really small. We can go through the size of things, and then they put little processes through these tunnels, and they're all touching. All these cells are touching each other in your bone right now, and they're repairing your bone, correcting cracks, micro cracks, things like that, in real time while you're just sitting there. But when you exercise, when you run, when you carry things, go upstairs, stuff like that, you put micro cracks in your bone, and these little cells are mechanosensors. They can sense the stress and the cracks in the bone, and then they automatically send out other cells to dissolve that part of the bone and then rebuild it. So they say your entire skeleton is replaced every 15 years by these little cells. So what we found though was this stretchy film that we peeled away from the inside of the horn, that first 48-inch long horn that we discovered in Glendive, Montana. And it was stretchy. And then when we thin sectioned it, we could see all the cells in it. So even the cells themselves are stretchy. So that was the first discovery that we made. That was a world first discovery. And then we found nerves were the first ones, and so far the only ones to find nerves with lipids, and not just in these Cretaceous materials, which is what we're mostly digging. That's where the dinosaurs are mostly found, in this uplifted Cretaceous bed around the world. It goes all the way around the world, but also in the Permian. We've gone into the Permian, which is accepted age of 300 million years, and even better lipids than we found in Cretaceous are found in the Permian, which is astounding. But we're the first one to find clots and identify them as clots in the canals. So in your bone canals, you have a vein, a vessel, and a nerve. They travel together throughout your body and on into your bones from the spinal cord. They go all the way out into your bones. They're called peripheral nerves. And so we found those, and we found clots in the canals. All the blood is clotted clear back to the Permian, and that suggests death by asphyxiation, drowning.
Mm-hmm.
This happens in drowning victims. When you drown and you die, your blood's all clotted. So it's a trauma that's inflicted on the system that causes the blood clotting cascade to initiate. And if it doesn't resolve itself, that is a one-way reaction that goes all the way to the end, and that's it for you. Now, often they revive you they'll revive these drowning victims in the emergency room. They'll get them breathing again, get their heart pumping, their blood circulating. And now, instead of clotting, because they burned up all their clotting factors, clotting everything, now they're bleeding out. So this is very common. It's throughout the medical literature. It's a well-known phenomenon. And so we surmise that these were all asphyxiated in one big event, all the way back to the Permian. And so that's a world first for us. And a lot of people are saying, oh, there's red blood cells in dinosaur bones. No, there are really no red blood cells left. They're all iron oxide or pyrite rhomboids, which are little spheres. And that's what we're confirming over and over and over again, that there's no red blood cells. There's just these iron oxide rhomboids that congeal into clots within the canal. So, the Bible says in Job, he's bragging a little bit about dinosaurs, isn't he? He's bragging, and he says, are not their bones like iron? Well, they're solid iron throughout the canals. That's why one reason they're so hard to break once they've been in the ground for a while, because all that clot is like rebar inside the bone. So, but we've thin sectioned a ton of these at different sites, different species, and they're all clotted. So, that's probably the most important discovery that we found, because it's like a snapshot of how they died, right? I mean, it's a permanent preserved in stone indicator that they were asphyxiated. And we've only found iron in the canals. It doesn't go out into the bone. So all the other tissues, the collagen, the cells that are out in the bone are not touched by iron. And you may have heard of this iron preservation theory. And in fact, I think we'll talk about that in a few minutes, but it's not in the bone. It's just in the canals. So what is preserving those cells and the collagen that's still out there? It's not iron, because they're not in contact with iron. So it's a very significant find. We're also the first ones in the world to find nematodes, which are worms that consume bacteria or bacteria biofilms, or they consume fungus. Fungi. We're finding fungi and bacteria in these bones. Why? Why are they there? Because there's tissue in there, and they're degrading it.
Mm-hmm.
And so we've also found nematodes in our thin sections. But lately, we have been recovering live worms, living worms, out of bone shards that we've scrubbed and scrubbed and scrubbed, and then we put them in a funnel with water. And after 15 minutes, the worms start coming out because they're drowning. And then we collect them and put them under the microscope. So we've actually sent those to PhD scientists who have identified them. That's a world first. I mean, it's shocking to know that living worms are in there. We're the first ones in the world to find insect remains in the bones. And we're publishing all this, and we're presenting it. Also, surface shards still retain tissues. Stuff that's been on the surface for a decade or longer still has soft tissue in it. Still has clots and nematodes. So you're generally not going to hear this from most of the sales organizations who do ministry, because they're not doing the work, so they can't sell it. And we've told them, please don't try to sell ours. But you'll hear that from Distry. We give it all away. This is for all of mankind to know. Another good thing that we're doing is our students. That, in a nutshell, is a set of the world first discoveries that Distry.org, the Dinosaur Soft Tissue Research Institute, has found and published in secular journals. We only publish in secular journals. And we're presenting. We go to the national meetings every summer, and we present this stuff. This year, our students are presenting these food webs that are inside these bones. There's food webs, living organisms consuming resources and working independently of each other, interdependently, inside these bones. So it's shocking.
Wow.
It is.
Yeah, a few things I wanted to go back and touch on that you mentioned is, first of all, it is just overwhelming how many things we are finding in these bones. So that's a lot. I did want to touch on the blood cells part. So I was under the impression that Dr. Mary Schweitzer had found blood cells in the T-Rex femur bone. Is that not correct?
If you go back and you look at her papers, her published secular papers, she calls them red blood cell-like microstructures, which is a more appropriate way to describe them, because number one, first of all, they're not oval. Reptilian blood cells are oval, and these are round. They're spherical. Number two, they're the wrong size. They're 10 to 100 times larger than what a red blood cell would be, even for a reptile. And so I think a lot of people misidentified them, and but we're documenting now that these are mostly iron oxide and sometimes iron pyrite framboids, or these little spheres that form as a result of just the interaction of the iron and the oxygen. That's rust, right?
Sure.
So this has a known chemistry that's all established in the chemical books and the literature for decades and decades. So, no, I don't think we're finding red blood cells. Nobody's really shown a compelling example of that. But if you look at all of her publications, she uses that terminology, and we followed that. In the beginning, we thought, okay, maybe they're red blood cells, but we didn't want to call them red blood cells. We didn't have the supporting information to really characterize that. But since then, we've found out these things, they glow in UV. We use UV microscopes to get the iron to react. It's called autofluorescence, and the iron will give off light when you hit it with light of a very high energy, UV. It's the same principle that when Moses came down from the mountain, they made him put on a veil, right? Because why? His face was autofluorescing from the energy of God as he was communicating with God up on the mountain. It put so much energy into his skin. Your skin will autofluoresce. I could take a piece of your skin and put it under the UV microscope, and it'll glow a lot of pretty colors. So that was actually a scientific phenomenon that was described in the Old Testament that people haven't really thought about. So,
Interesting.
but that's all we're doing. We're taking advantage of that property of the matter to hit it with a unique wavelength of light, and it comes back and says, "Yep, I'm here." So we have positive identification that it's pooled iron and not hemoglobin. Now, Dr. Schweitzer did find remnants of hemoglobin that have signature for reptilian and avian. They often say, the birds, or one segment of the birds, came from the bird-like dinosaurs. But no, we've proven pretty conclusively that these are iron oxide framboids and not red blood cells.
Okay. Well, thanks for that clarification. The other thing I wanted to ask about is, you're talking about extracting these, films and things being stretchy and stuff. How exactly are you doing that? Maybe in a nutshell, for some of us who maybe aren't all that familiar with microscopy, how exactly are you going about that?
Yeah, good question. And anybody can do this. We have a philosophy called simple man science. And for example, our president, Keith Holcomb, has had a long career as an aviator. He's a captain for one of the big airlines, and he flies out of Dallas. And he's being forced out of his career because at 65, pilots, they're cut loose, right? So he's coming up against that, but he's already learned enough microscopy that he could work in a lab right now in a nice career. But the one thing we want people to understand is that anybody can do this. You can collect dinosaur bones yourself and bring them home and put them through a decalcification protocol, which is just a weak acid like a vinegar that you soak it in, and then you can pipette out the cells and the veins and the nerves and all that. But as far as the stretchy stuff, one of the things we do is we immediately put our bones at the site right out of the ground. We put them into fixative called formalin. A lot of people recognize it as formaldehyde, but it's a 10% solution of that, and we put the bones in there immediately, and so that kind of makes the tissues a little more resilient, so you can stretch them and they won't rip or break. And so when we fractured open the horn, we just use our hands. It was full of plant roots. It was full of fungus with big white fungal maps in there. When we opened them up, the whole bone, the horn broke into about 4 or 5 different pieces when we took it out of the ground. Even though we jacketed it, with the plaster Paris jacket and all that. Mm-hmm. So we were able to just put mechanical pressure on it with our hands and break it open. And as we did that, there was a core. You could see this sort of round, pillar cylinder in the center of the horn. That was the horn core, and on the face of that was this flap of tissue that we just started peeling away from the bone. You can see that in a video. We have a YouTube channel, and I'll give you the way to go to that. But all this stuff is up there. So the formalin, pardon me, itself, the fixative itself, kind of stabilizes the soft tissue. And if it is inherently stretchy, it will be even more stretchy and will not tear as a result of putting it in the formalin. So the other advantage of putting our specimens in formalin at the site is we capture the living nematodes and freeze them in place, and all the other structures are frozen in place, so that when we thin section them or decalcify them, which just means it's like dissolving them in a dish, but all the tissues remain, so you can pluck those out. But that kind of is the process. It's not a lengthy process. It might take you, about a month just to put them in the formalin, and you want to wash them, and we air dry them, and then we'll put them in the weak acid to decalcify them and pipette out whatever we want to get out of there off the bottom of the dish, or we impregnate them with a polymer, a plastic that hardens very hard in the oven, and then we can thin section them into thin sections that are half the width of your hair. So they're very thin. You can see all the cells in there. You can see some of the soft structures, but a lot of those are transparent. So they don't show up that well in the thin sections. But in the canals, we can see all the nematodes and all the framboids, and whatever was preserved is still in the canals. And we train you to do this. We train people, anybody, to do this. In fact, we have students where we have, and they're in different states. We actually set them up with a microscope workstation. We go there and we train them on how to do their own research. We give them a project with a certain set of bones, and they get their own results. They photograph them. They have a camera on their microscope so they can document everything, and then we get them published. And so we have a 17-year-old now who's got 2 papers that he's already published, and he hasn't even finished high school yet. So anybody can do this. It doesn't take someone with a PhD, and that's our point. That's why we love bringing in new students, so that they can do this on their own, discover the science behind it, and learn it, and then go present. So our students are going to the International Microscopy Society of America meeting in Salt Lake City in end of July, and they're going to stand up and teach the PhDs what they're finding. So it's really pretty cool.
Yeah, that is amazing. Yeah, I can tell you're an educator at heart and really have a passion for reaching the next generation.
Absolutely.
Moving on to maybe some of the responses from skeptics whenever your findings are presented and what have you. What are kind of some of the main objections that skeptics will put to saying you're not really finding what you think you're finding?
Well, to be honest with you, that's not really the response we get. We do get people who frown when they see our posters or hear our platform presentations, and they might ask questions. And generally, their questions have been unanswered, and so we can answer their questions. And so it's amazing, Preston, because we're building relationships with scientists. We had 3 PhDs come to our Glendive dig this year from universities in Texas A&M, and a couple of others. And they've invited us, come to our lab, bring your specimens, and we'll play with our $500,000 to $1 million microscopes, which is really cool. So no, we're actually building bridges, and they made me an editor on the journal for Microscopy Today, and so it's really kind of a flowery title, but it means I have to work a lot more. So I have to produce 3 or 4 papers a year. Sure. But with the students, we can do that, so I don't have to do all the work myself. But we're building collegial relationships because we don't talk about the controversy. I mean, everybody knows the controversy, right? So why delve into it? It's better to find out what is really comprising these tissues and how much have they been altered, if at all. And they are altered a little bit, but many of them not significantly. So it's really quite interesting. It's people know us as those dinosaur folks at the meeting, and they'll come up and ask questions. But we also have another reaction. We had one gentleman who very high up in leadership and for the society, and he was at a big university in the Midwest, and he heard that we had a nerve slide. We had a slide with a nerve on it, and came and asked if he could see it, and said sure. So a little bit of trepidation, I gave it to him, and then he took it away that night. But the next morning he came up and he was visibly shaken and irritated and upset and frantic, and then the poor guy looked really afraid to me and made me take it back and told me, I can't do this. My microscope won't work. Or, he was just babbling things. And so I think some people are really so shocked by it that they fear being associated with it. But generally, most of the folks that we deal with are, they're very open. They have good questions. They have great challenges for us to improve our science. So we're growing. as they're learning. So it's really an amazing thing. And so we don't present the controversy. We don't discuss it, and we come across as collegial and friendly and approachable, and it's working.
Wow,
that's great.
Yeah, I'm glad to hear that we're making those bridges that way, because really, if you're doing real science, then there really is no reason for people to get defensive over worldview issues, because we're just talking about the science here, and let the implications be what they are. But let's do good science.
Well, yeah, and if I'm wrong, I want to know. I don't want to be out there doing bad science. But so far they're like, "Yep, no, green light. You're doing good." So, it's really a neat thing, and the opportunity, I think, for students. A lot of students don't know how to get into science. They love science, but oh gosh, I got to go do a whole bachelor's degree and then maybe a master's. No, you can learn these skills, get into a lab on campus, and start working, earning good money with benefits, and then you can go to school while you're doing it. That's exactly what I did, and I had a wonderful 50-year career in microscopy, and just so much fun, so exciting. Every day it was so fun to get up and go do something new with microscopes, and so those careers are available really with the training that we offer, the skills that you will learn by publishing. And when we go to the meetings, the professors are looking for students like this. They want to matriculate them in their own programs and let them run their labs. And so there's just a lot of opportunity to get into a great scientific field. And so many things are affected, by microscopes. I mean, just look at Pasteur and what he did with microscopes. He saved the wine industry, the tanning industry, the wool industry. I mean, over and over and over again, this guy turned his microscope to problems that they were having in France with all these different industries, and he saved them all with the microscope. So that hasn't ended. So there are really cool positions available where you can really learn. You can get into a doctoral program if you want, which is what I did, and go into teaching or research, or there's just so many opportunities. So it's really amazing to me that this is happening.
Wow.
Yes, that is amazing, Dr. Mark. That's the way to go, financially and for the career.
Yeah.
Yes, for sure.
Yeah.
Well, all these things that you've mentioned that you find in these bones, they've just been blowing me away this whole conversation. I had no idea a lot of these things, but ultimately, why is this important, the things that we find, and what can we learn from the things that you're finding?
Right. Well, of course, and I'll touch on the iron preservation theory now because I think it's relevant. Dr. Schweitzer proved without doubt that these are original tissues, that they do go back to the dinosaur. She really did a whole series of elegant experiments that really nailed it down. And so it cannot be contamination. It's not something left over from the bacteria or the fungus. It's really the original tissue that was in the bone from the beginning. And so she had to come up with an explanation for why these things are still here, because we know that tissues don't last that long, especially just sitting in the soil. With a mummy, they've covered it with all kinds of spices and things, and then wrapped it very tightly and stored it in a unique environment. But not so with these bones. They're sitting in wet sand for how long, class? That's the question, isn't it? How long have they been sitting there, right?
Right.
And so by looking at these things, we can really make good interpretations that these can't last for long ages. So some kind of preservation mechanism is required. And that's what Dr. Schweitzer did. She came up with what might seem on face value to be a logical answer, and that is all the iron that was in the hemoglobin of the blood somehow became free and spread out through the bone. And when this naked iron touches cellular architecture or tissues, and this is all in the medical literature, normally it rips it apart like Pac-Man. So it's naked iron in the blood is a very dangerous thing. It really tears up organs, all kinds of tissues, and can lead to death. And there are people suffering from it. I was talking to a lady not long ago who had inhaled a broken capsule of iron that she was taking, supplementally, and she breathed it into her lungs. Now her lungs are burned to smithereens, and she barely walk around without. And she's young; she's in her 40s without breathing heavily. So the iron, under normal circumstances, destroys tissues. It does not preserve it, but it will, according to their paradigm, twist things just enough to render it a little bit more resistant, let's say, to decay. That's the thesis. And so, but by studying what we've studied, we found not only is the iron not free, it's bound up in these clots and it's in the canals, but it has not gone out into the bone to preserve anything. So there still is no preservation mechanism for this, which astonishes me, because I can look at slides that I made 3 decades ago and they've deteriorated, even though I went through all the protocol. They're still deteriorating. And so to find these cells that are beautiful, they have all these little filopodia around them, and you can see all this on our website. We've got pictures, and all the papers are there. You can download the papers. You can see these structures, the nerves with beautiful crosshatch pattern, and lipids. I mean, the lipids ooze out of these things. That should not be there. The lipids should have decayed away to dust a long time ago, if they've been there for 3 or 4 or 5,000 years. So it really puts an outer age on these tissues at about 10,000 years. They really can't go much longer than that. And of course, the conventional geological paradigm is that millions of years are involved in this. And to have living worms eating the fungus, which is eating the tissue inside these bones, is shocking. So it puts an outside limit on it. And so it forces us to really grapple with the age of the earth and all of these layers. It really forces us to look at it. And so it can be quite controversial. Often we will say in our papers, this is controversial. We admit that because a lot of people still cannot believe that they're there, but they are. And I made a prediction in 2012 that this would be the exception. The norm instead of the exception. And it's turned out to be that way. This is the norm. We're finding it in everything. So most people are under the impression that most dinosaur bones are hard rock. And they're not. They're just like, if you go into a hospital and you have to give a bone specimen and a biopsy, they'll put it through the same protocol that we're using to decalcify the bone. So we're not reinventing anything. We're just doing what hospitals do. So they respond. They take up stains, all the standard laboratory stains that are on the laboratory shelf in a microscopy lab. They take them all up. And so we've even stained them with acridine orange, which shows RNA, DNA, and they light up. So there is RNA in there for sure. But a lot of it's degraded. But no, they're behaving as normal tissues would. So,
wow.
That shouldn't be, right? But it does make you really sit back and think about, when did these things happen? What was this? Because it's all sedimentary. Was this a global flood? And the answer keeps coming back. Yeah, probably every time. So, yeah.
You mentioned DNA and RNA being discovered in these bones also. I kind of like to touch on that because it's my understanding that the half-life on DNA is, right around like 520 years or something.
Yeah,
so
exactly right.
Have you been able to, or I guess maybe I should ask, how much DNA are we finding in these bones, and does that have any bearing on their being able to detect their age?
Well, not really, because DNA is a sugar molecule. It has a sugar backbone, the ribose, deoxyribose nucleic acid. So that gets gobbled up pretty quick by the decomposers that are in there. And we're finding microbes that would be like pond water organisms, plus the nematodes, plus bacteria and fungus. 3 different kinds, or maybe 4 different kinds of fungus we've found so far growing in these guys. So they're under heavy attack. And that just again speaks to the fact that there's stuff in there that shouldn't be in there, that couldn't have been washed in. I mean, you don't wash in soft tissue in the bones, right?
Right.
But And the canals are so tiny. I mean, they're you have to use a microscope. All this stuff is microscopic.
Mm.
So you have to use good microscopes to look at this and image them. But like I
Mhm.
said, simple man science, anybody can do it. And we've proven that over and over. But
Mhm.
no, there's no preservation mechanism that we know of. And so how long, class, can these things last in the soil? That's the question. And it certainly can't be over 10,000 years for sure.
Wow.
Yes, that points us back to our authority as
Yeah.
Christians, the Word of God, the infallible Word of God, where it says in Genesis that God destroyed the entire earth with
Yeah.
water. And what would we
Yeah.
expect to find if the world was covered over with water? And
Yeah.
we do want to be consistent with God's Word. We find that we don't have to, play a part. We don't have to try to fit this in here and fit that in there, and this works together. God's Word stands on its
Right.
own accord.
Yeah.
And
Yeah.
Dr. Mark, I'm privileged that we do share that same belief, and we would love to just hear that life-saving message that
Mhm.
that Christians all around the world have been believe in for their source of joy of life,
Mhm.
eternal life. And would you mind sharing that wonderful gospel message with us?
Yeah. No, this has strengthened my faith. I mean, and I believe, I really believe that God, I often ask, why me, God, or why district? I mean, we're nobody, basically.
Mm.
We're just a ragtag group of people who are just doing the work, and it's showing one new thing after another. And why me? And I think it's because early on in my walk with the Lord, I decided your word, God, is going to rule my life, whether I do well at it or I do poorly at it, but I'm going to grow in it. And so I think because instead of saying, show it to me, God, prove it to me, God, and I'll believe you, I said, I will believe you, God, and he's proving it to me.
Mm.
So I think having that kind of faith is what he rewards greatly. I never dreamed that, I would be involved in this work and making such world discoveries and having students do it also.
Mm.
I just thought I was going to be a lab tech enjoying microscopes for the rest of my life. But I think he chose me because I do trust his word and I don't question it. I may not understand it,
Mhm.
but if I give it time and I give it thought and I give my heart to it, to ponder it, it says Mary pondered all these things in her heart, didn't it? It
Mhm.
says that. So that's what we do, and it's a walk. You get better at it every day,
Mhm.
and I think he rewards that. He rewards our faith by doing amazing things, and I think in my case, he's certainly done that. So
Mm.
I would say to anyone out there who's on the edge, just trust him. Just go ahead and trust him. What's it going to cost you, really, to trust him, to really, really trust him? Probably not much, but what is it going to reward you with?
Yes.
Um,
Mhm.
he says in the Sermon on the Mount, he's talking about teaching those who teach and practice these things, he says, right?
Mhm.
And he says they will be called great in the kingdom of heaven. But that's really a poor translation. It really means we'll be called to greatness in the kingdom of heaven. We'll be invited to greatness is really the actual word. So he does reward us when we trust him and we put if trust is your currency, God will cash your check every time.
Mm.
It's a very good way of putting that. So,
Mhm.
Dr. Mark, we just thank you for joining us today on the podcast. And where is the website where people can learn more about your work and
Yeah.
your research going on and that sort of thing?
Excellent question. So we took the first letter from each of the words in Dinosaur Soft Tissue Research Institute. So dstri.org. But the free books are there, all the papers are there. And this is an open invitation to whosoever will. I mean, if you're interested in a career in science, you can contact us through distry.org and let us know, and we'll start the process. And we also have videos on distry.org, but we have a YouTube channel. If you go to YouTube and in their search bar, you type in Mark H. Armitage. So our channel will come up and you can see all the latest videos on things that we've been discovering at the digs and stuff like that. So,
well, we're grateful for the investment that you guys are making and that you are the ones who are doing this work. The rest of us are very appreciative for this kind of research and everything that you're doing. So thank you again so much.
You bet. Thank you guys so much, and God bless your audience.
Folks, we encourage you to visit the website, dstri.org, and we also would encourage you to visit our website, the creationexperiencemuseum.com, where you can learn about our museum here in Branson, and you can learn about the free guided tours that we offer Wednesday through Saturday from 10 a.m. to 5 p.m. Last tour of the day begins at 3:30 because we want you to be able to see everything before we close up. So feel free to visit us, email, call, follow us on social media, and tune in next time for our next podcast when we will be continuing to showcase the beauty of God's creation. This podcast is produced by the Creation Ministries of the Ozarks, which also operates the Creation Experience Museum in Branson, Missouri. If you're in the Branson area, come visit us and get a free guided tour and see how science confirms the Bible. You can book your free tour at creationexperiencemuseum.com or call us at 417-561-0750. We encourage you to follow us on Instagram and Facebook. On behalf of our entire team, I'm Preston Knapp, inviting you to join us again next time as we continue to explore the marvels of God's creation and grow in our love for Christ.